Okay, Cardiovascular Physiology, Chapter 14. In this chapter, we're going to talk about the cardiovascular system and look at the importance of blood pressure and fluid volume on blood flow and resistance to blood flow. We'll look at the heart muscle itself, so our cardiac muscle and how it works, and then just take a look at the heart as it functions as a pump. So here's an overview of the cardiovascular system. So let's just follow blood. We're going to start with deoxygenated blood returning to the heart. So we have our superior and inferior vena cava bringing deoxygenated blood into the right side of the heart. It will dump into the right atrium. From the right atrium, it'll go into the right ventricle through our AV valve, and this AV valve is our tricuspid valve. From the right ventricle, it'll get pumped out into the pulmonary artery. Now, it'll go out through our pulmonary veins. The pulmonary artery is the only artery in our body that carries deoxygenated blood. That deoxygenated blood will then go to our lungs where it will get reoxygenated. The oxygenated blood will then return back to the heart. Now, it'll return back to the heart through the pulmonary veins, and the pulmonary veins are the only veins in our body that carries oxygenated blood. And it'll bring the blood back to the left side of the heart, in this case, the left atrium. From the left atrium, it'll go through the other AV valve, which is your bicuspid valve, also known as your mitral valve, into the left ventricle. And when the left ventricle contracts, it'll then send it out through our aortic valve into the aorta. And by the way, the pulmonary valve and the aortic valves are also known as your semilunar valves. And when blood goes out the aorta, it will then go to our body to feed our body oxygen, so to oxygenate our tissues. And when our tissues use oxygen, it uses it to make energy, right? It does aerobic metabolism. We will pick up carbon dioxide, and then that oxygen low blood will then get sent back to the right side of the heart. And then the cycle just continues over and over again. Oh, I wanted to point out really quickly, here is a portal system. So remember, a portal system is where we connect one bed of capillaries to another bed of capillaries. So we learned about our hypothalamic hypophyseal portal system when we looked at the endocrine system. Here is our renal portal system in our kidneys and our hepatic portal system that connects the liver and the GI tract. And we'll talk about that when we get into the digestive system. So pressure is important for blood flow, because blood only flows down a pressure gradient. So blood only flows from high to low pressure. If there's no pressure gradient, if there's no difference, then blood flow would stop. And so that's why that left ventricle is so important, or the heart in general. But again, we really focus on that left ventricle because that's the one that sends blood out to our body. So the left ventricle is going to create the greatest amount of pressure. As a result, the aorta has the highest pressure. And as blood flows from our aorta to our arteries, to our arterioles, capillaries, venules, veins, vena cava, and back to the heart, notice blood pressure drops. Now, pressure is created by the contracting of our cardiac muscle. And like I said earlier, the ventricles are going to be the main creators of that pressure. And just so we understand, if blood vessels constrict, when we vasoconstrict, that will increase blood pressure. When we vasodilate, that will decrease blood pressure. So when we want to change where blood flows in our body, we will actually constrict or dilate our blood vessels. And changing the radius of our blood vessels actually impacts blood flow the greatest. So it has the most influence because it has an influence to the power of 4. So it's exponential. Volume changes can also affect blood pressure. So when we increase blood volume, blood pressure increases. When we decrease blood volume, blood pressure decreases. So this is just looking at the influence of pressure on blood flow. And again, it's all about the pressure gradient, which means the difference. So here we have a tube with 100 millimeters of mercury pressure on one side, 100 millimeters of mercury on the other. And so there will be no blood flow here, or no fluid flow here, because there's no difference. In this image, we have 175 in this tube and 40 and 15 in this tube. Now, although the absolute numbers are different, the difference between the two sides are the same. So this is a difference of 25 millimeters as well as over here 25 millimeters of mercury. And so even though the absolute numbers are different, because the difference in pressure is the same, you'll have equal flow between both of these tubes. So it's all about the gradient. Now, I mentioned earlier that the radius is going to have the greatest impact on blood flow resistance and as a result blood flow. And so the bigger the radius, the greater the blood flow. And you can see that here. And that's really because radius has a relationship to blood vessel resistance and as a result blood flow to the power of 4. So it's an exponential relationship, which basically means small changes in radius have a really big impact on blood flow. So let's take a look at our heart. Again, this is more anatomy, so I'm going to go through this fairly quickly. But here again is our vena cava, superior and inferior, bringing blood into the right atrium. It'll go through the tricuspid, our AV valve, into the right ventricle, which will then pump it out our semilunar valve. This one would be the pulmonary, into the pulmonary arteries. Then the pulmonary veins will bring blood back into the left side of the heart through the bicuspid, also known as the mitral valve, our other AV valve, into the left ventricle, and that will then send blood out through the aortic valve into the aorta. So if you are unsure about blood flow, this is just a bigger diagram that shows what's going on in the heart and how the blood valves are, where they're located. Okay. All right, so let's take a look at heart muscle, cardiac muscle, and we'll compare it a little bit to skeletal muscle. So first of all, cardiac muscles are smaller than skeletal muscles. So skeletal muscle fibers, remember, they're really long. Cardiac muscles are not so much. Also, whereas skeletal muscles were multinucleated, cardiac muscles are not. They have a single nuclei per muscle fiber, and it's located centrally. But cardiac muscle has this structure here called intercalated discs, and these are your intercalated discs. And it's really important because you find two important junctions here. One are your desmosomes, and these are going to allow force to transfer from one cardiac muscle cell to another. The other really important one are your gap junctions. So gap junctions we learned about during our first week, but they are connections between two cells, but there's an actual hole or an opening. And so whatever happens in one cell can quickly move to and influence the next cell. And in this instance, it's going to be the depolarization, the electrical impulses that get transferred from muscle to muscle. What else? So T-tubules are larger and they branch more here. And the sarcoplasmic reticulum is smaller. And one of the reasons is because in cardiac muscle, we also rely on extracellular calcium from the extracellular fluid. So we're not just relying on calcium from the sarcoplasmic reticulum, which is the case in skeletal muscle. And in cardiac muscle, 1/3 of the muscle cell is occupied by mitochondria. And that makes sense because mitochondria is our powerhouse. This is who gives us energy. And we really never want to run out of ATP and fatigue in our cardiac muscle. So let's take a look at excitation-contraction coupling in cardiac muscle. So who stimulates or controls our cardiac muscle? That would be our SA node. So the SA node will depolarize and fire an action potential, and then that action potential will travel from muscle to muscle through the gap junctions. And so here's our action potential traveling along our cardiac muscle. Now notice this figure looks a lot more similar to skeletal muscle than smooth. And you're going to see a lot of similarities, but there are important differences, so make sure you note them. So the action potential is going to travel along the sarcolemma and it will go down our T-tubule. And that's similar to skeletal. And then we have our voltage-gated DHP receptors, our dihydropyridine receptors. Again, similar to skeletal, but here's the difference. In skeletal, the DHP receptor just changed shape. It didn't actually open and allow anything to flow through it. That's different here in cardiac. This voltage-gated DHP receptor, when it opens, actually allows calcium to come in from the ECF. And when calcium comes in from the ECF, it triggers the ryanodine receptor to open so that more calcium can come out of the SR. So here's the difference between skeletal. In skeletal, this ryanodine receptor was mechanically gated because it was physically connected to the DHP, but not in cardiac. In cardiac, this ryanodine is chemically gated because the calcium chemical is what's triggering it to open so that more calcium can come out of the SR. After this point, everything else is the same as skeletal muscle. So calcium will bind to troponin because our cardiac muscle is also striated. It has sarcomeres. And when it binds to troponin, it'll cause troponin to undergo a conformational change. So troponin will change shape, which will move tropomyosin, and now the myosin and actin can form a cross bridge and do its power stroke. And then a new ATP comes and binds to release, and we have our contraction happening, similar to skeletal. Now to relax, we have to put calcium back where it came from. So in this case, calcium came from the SR, so we're going to pump it into the SR. It also came from the ECF, so we're going to take it out into the ECF with this sodium-calcium exchanger. And as we get rid of calcium, calcium will unbind from troponin, which means tropomyosin will get moved back to its original position. So the myosin binding site on the G-actin molecule will now be covered by tropomyosin, and so cross bridges will not form, and so your muscles will relax. So cardiac muscle contractions can be graded, just like skeletal, meaning we can have a strong contraction, we can have a weak contraction. And the length of your sarcomere will affect the force of contraction. Again, just like skeletal, because cardiac muscle also has sarcomeres. So when your sarcomeres are too short, you'll produce very little force. When they are too long, you'll also produce very little force. When your sarcomeres are too short, you'll produce less force because you won't have a lot of room to slide. When your sarcomeres are too long, you'll produce less force because there's not enough overlap for cross bridges to form. So we do have an optimum sarcomere length. And action potentials also vary here. So we're actually going to learn two new action potentials. So the first action potential I want to go over is to review the action potential we're already familiar with, which was the action potential that occurs in our neurons, the ones that occur in our skeletal muscles. And that's the one where we have that rapid depolarization due to sodium rushing in, and the rapid repolarization due to potassium rushing out. So that's the one we're familiar with. Here is our new action potential, and this one is the one that's generated in our cardiac muscle. And so here's what happens. We have a rapid depolarization still due to sodium rushing in. So this P means conductance. So that means we have high movement of sodium. Okay, so sodium is rushing into our cell, causing that depolarization. Now sodium permeability has reduced, meaning it's not moving anymore. And then typically we would just have potassium starting to move. Now potassium starts to move slowly, right? The permeability is still a little bit low, but what I want you to pay attention to is that now calcium is able to move. So remember, calcium is a big player in all muscles. So what happens is potassium starts to move into the cell, but at the same time calcium starts to move out. So if potassium is starting to rush out of the cell, and then calcium starts to rush, wait, let me do that again. Okay, so let me make sure I think I said something wrong there. So let me reiterate this. So sodium rushes in, that causes our depolarization. Potassium starts to move out, but while potassium is moving out, calcium is rushing in. Okay, so there we go. While potassium is moving out, calcium is coming in. So what we have is pluses going out and pluses coming in. Because we have pluses going out and in at the same time, we get this plateau here. Now this plateau is going to be really important, and we'll talk about that next. Then what happens is calcium movement stops, and then more potassium channels open, and potassium continues to rush out. And as a result, we get our repolarization. So let's do that one more time. So the depolarization is due to sodium in. Then we start to move potassium out, but while we move potassium out, calcium is coming in. So pluses out, pluses in means we have essentially no change in our membrane potential. That's why we have the plateau. Then calcium movement stops and potassium continues to flow, and it flows much greater. So potassium rushes out, and that potassium moving out causes the repolarization of our action potential. So this is the action potential that occurs in our cardiac muscle. So why is the action potential different, and why is it important? So here's why. So let's take a look at skeletal. So here's our skeletal action potential. So sodium in, potassium out. It's a very short, quick action potential. And then our skeletal muscle will respond. It will contract and then relax. And so what we can do is, as soon as the action potential is over, we can actually give another action potential and another. And that's what's happening here. Every one of these little triangles is an action potential. So as soon as the action potential is over, while the muscle is contracting, we can give another and another and another. So the contractions will sum and add on top of each other. And if we do that enough, we reach tetanus, where we have sustained contractions. And that's okay for skeletal muscle. But let's take a look at cardiac. And if you just think about our heart, the job of the heart is to relax and fill up with blood and then contract and eject blood. So we don't ever really want our heart muscle to contract and sustain that contraction. That means we're going to contract and pump out blood and then just stay contracted. We don't want that. We want it to contract, and then we always want it to relax and refill. Contract, pump out blood, relax, and refill. And so to prevent the cardiac muscle from ever being able to sum and reach tetanus, we have this long action potential. So again, the action potential is due to sodium rushing in, then potassium coming out and calcium coming in at the same time, and then potassium going out would be the repolarization. Now notice, as soon as your cardiac muscle action potential is over, the cardiac muscle contraction is also over. So even if we were to give another action potential as soon as possible, so as soon as our heart muscle's action potential is over and we give another one, because the contraction is also over, we'll never get summation. And if we can't sum our contractions, we won't be able to reach tetanus. And so it helps protect our heart from reaching tetanus. So it's a good thing. All right, so here's our second new action potential. And our second new action potential is found in our SA node. So our SA node is our pacemaker. It's going to control our heart rate. So really focus on this middle figure here. So it's all about calcium and potassium. So the depolarization phase of the action potential in the SA node is due to calcium rushing in, and then the repolarization is due to potassium rushing out. So calcium in, potassium out. So we have our three action potentials. The one that you knew, sodium in, potassium out. Then the new one in the heart muscle, which has that calcium and potassium moving at the same time, causing that plateau. And then this SA node action potential. All right, so our SA node contracts autorhythmically, so all by itself it will contract, but we can certainly influence our SA node and our heart rate, and our autonomic nervous system will do that. And so the cardiovascular system is controlled by our medulla oblongata, and depending on what we need to do, we will either activate the sympathetic or the parasympathetic nervous system. The sympathetic nervous system speeds up heart rate, parasympathetic slows it down. And the sympathetic will speed it up by releasing the neurotransmitter norepinephrine. And when we release norepinephrine onto the SA node, it basically causes us to reach threshold quicker. So here's in gray our normal SA node. This is what it would normally beat like without any influence. And when the sympathetic releases norepinephrine onto it, we reach threshold quicker. So notice the action potential itself doesn't change size, but we can depolarize quicker to threshold, and that's how we increase heart rate. If we want to slow down heart rate, we'll activate the parasympathetic nervous system. That releases the neurotransmitter acetylcholine. And what that does is it hyperpolarizes our membrane potential a little bit. And so when you hyperpolarize it, you're further from threshold, so it takes you longer to reach threshold. And as a result, your heart rate slows down. So again, the action potential itself doesn't change, but how long it takes to reach threshold does. All right, so again, just so that it's clear, our two new action potentials, we had the one in the heart muscle itself with that plateau, which we said is due to the movement of calcium, and then we have the new action potential in our SA node. And so when our SA node fires, only the muscles that are right next to it really get depolarized, but that depolarization and the action potential will travel from muscle to muscle through the intercalated discs, which we said have those important gap junctions. Okay, so the SA node, again, is our pacemaker. It's set to beat our heartbeat at about 70 beats per minute. The AV node can also be autorhythmic, so it can kind of take over if we have any issues with the SA node, and it beats at about 50 beats per minute. And then the Purkinje fibers beat at about 25 to 40 beats per minute. And we can measure the electrical activity of our heart. And we do that with Einthoven's triangle. And this is called an electrocardiogram, an EKG or an ECG. And basically, it gives us indirect information about heart function. And if you've ever seen an ECG recording, this might look familiar to you. We can name the different waves, and they represent different things. So we have our P wave, then we have our Q, R, S complex, and then our T wave. So this is one cardiac cycle. So let's take a look at what they represent. So I'm only going to focus on these major waves. I'm not really going to focus on the segments and the intervals. So let's go over the P wave. So the P wave represents depolarization of the atria. So an easy way to remember it is, it's where your atrias are going to contract. The QRS complex represents depolarization of your ventricles. So you can think of it as your ventricles are going to contract. And then the T wave is repolarization of your ventricles. So you can think of that as your ventricles are going to relax. So after a depolarization, we need a repolarization, and we see that for the ventricles. But we didn't see it for the atria. So here we have our atrial depolarization, but nowhere do we see atrial repolarization. And that's because it's hidden. So while the atrias are repolarizing, the ventricles are depolarizing. And since the ventricles are bigger and the depolarization is stronger, it's essentially hidden within this QRS complex. So they do repolarize. We just don't see it separately. Okay, so let's take a look at a cardiac cycle. That is one contraction relaxation. So systole is when the heart is contracting. And when the heart is contracting, we're really talking about the ventricle. So it is pumping out blood. And then during diastole, the heart is relaxing and it is filling with blood. Just to define more terms, stroke volume is the amount of blood that's pumped out every time your heart contracts. So every time your ventricles contract, the amount of blood that's getting pushed out is your stroke volume. End diastolic volume is the volume of blood in the heart at the end of diastole. So we said diastole is when the heart is relaxing, and this is when the heart is filling with blood. And when we say heart, again, we're talking about the ventricles. So when the ventricle has finished doing diastole, finished relaxing and filling, the volume of blood that's in that left ventricle or in the ventricles is your EDV. End systolic volume means the volume of blood in the heart at the end of systole. So after that ventricle has contracted and pushed out blood, there's still some blood left over in the heart. That amount is your ESV. So if we want to know how much blood is getting pumped out every time your heart contracts, we can take your EDV, which is how much blood is in the heart when it's completely full, and subtract ESV, which is the amount of blood that's in the heart after it's contracted. And if we subtract the two, we'll be able to find out how much blood actually went out with each contraction. That's your stroke volume. Cardiac output is the amount of blood that gets pumped out of the ventricle per minute. So we're going to let the heart contract for an entire minute and see how much blood is getting pumped out of the heart. So to figure out cardiac output, we can take our heart rate, which is how many times your heart contracts, and multiply it by our stroke volume. And we know stroke volume is EDV minus ESV. And the average cardiac output is about 5 liters per minute. And when you talk about the heart, you might also be familiar with the heart sounds, lub-dub. So lub-dub is the closing of your AV valves. Sorry, I don't know why there's 2 V's. And then dub is the closing of your semilunar valves. So lub-dub. All right, and then stroke volume. So stroke volume, we said, is the amount of blood that gets pumped out of the heart every time your heart contracts. So obviously, how forcefully your heart contracts will influence stroke volume. So when we increase the contractility of the heart, contractility means how forcefully your heart is contracting. So if we increase the contractility of the heart, your heart is contracting with a lot of force. And so if it's contracting with a lot of force, it's going to pump out more blood. So higher contractility means greater stroke volume. Now, if you think about that, if we pump out more blood, there's going to be less blood left over after the contraction. So that means ESV is likely to be lower. So hopefully that all makes sense. Then we have something called the Frank-Starling law. Okay, so the Frank-Starling law is where the this is where the sarcomere length comes into play. So remember how we said there's an optimum sarcomere length, but your heart muscles function shorter than optimal. And I mentioned that during the cardiac lecture. So the reason your heart muscles function shorter than optimal is because when more blood returns to the heart, when we increase our end diastolic volume, remember, EDV is the amount of blood that's going to fill the heart or be in the heart when it's completely full and relaxed. When we increase that, that means our heart is getting stretched. And so the more we stretch our heart muscles, the more our sarcomeres are going to get stretched. And what we want is we want the stretching of our sarcomeres to cause our sarcomeres to contract with more force. And so we want our sarcomeres to function shorter than optimal so that the more we stretch, the closer to optimal we get, and so the more force we will produce. And so that's our Frank-Starling law. So stroke volume will increase as our EDV increases. So as we bring more blood back to the heart, our heart will automatically contract with more force. It'll automatically increase contractility, which will increase our stroke volume. And so what determines how much blood comes back to the heart? Well, that would be our venous return. So the sympathetic nervous system can influence that by constricting our veins. And by constricting our veins, it's going to push the blood into the heart. And our skeletal muscles and our respiratory muscles can also help with that. So when our skeletal muscles contract, it's going to help squeeze our veins and push the blood back to the heart. And when our respiratory muscles contract, it's going to push it back into the heart as well. Okay. And an inotropic effect. An inotropic effect is any chemical that affects contractility. So norepinephrine, for example, will increase the contractility of the heart. It will just make the heart beat more forcefully. Digitalis is a chemical that's given to also increase the contractility of the heart. So at every EDV, so at every length of sarcomere, this chemical will just cause your heart to beat with more force. And so this is a good summary slide. So cardiac output is the amount of blood that gets pumped out of the heart per minute, and we said it is a function of, or it is heart rate times stroke volume. So this shows all of the things that can influence heart rate. We know it's autorhythmic cells that control heart rate. That's your SA node. But the SA node can be influenced by the parasympathetic and the sympathetic nervous system. So the parasympathetic can decrease heart rate. The sympathetic can increase it. So if we activate the parasympathetic nervous system, which we know decreases heart rate, that will decrease cardiac output. So this is how you can kind of use this figure to help you really connect the dots. Now, what about stroke volume? So lots of things influence stroke volume, the amount of blood pumped out per contraction. So it's determined obviously by the force of contraction. So we said the greater the contractile force, the greater the contractility, the greater the stroke volume. So contractility can be influenced by the sympathetic. So the sympathetic nervous system releasing, for example, epinephrine onto the muscle can cause it to contract with more force. Also, the contractility of the heart is influenced by our end diastolic volume. So remember, the more blood that comes back to the heart, we're going to stretch our sarcomeres, and when we stretch our sarcomeres, it's automatically going to contract with more force. That's our Frank-Starling law. And again, that's all about sarcomere length. And EDV, the amount of blood that gets pumped back to the heart, is influenced by things like our venous return. So whether our blood vessels are constricting to push blood back into the heart. And then we said our skeletal muscles and respiratory muscles can help aid that. So this is a good summary slide. Okay, so thank you for this chapter.